Okay, guys, we are going to start moving on with cell respiration. Remember, we just did glycolysis. So from glycolysis, we are going to get pyruvate. And that's going to go to the second step of cell respiration, which is the prep reaction. So the prep reaction, it is going to prepare pyruvate that we just got from glycolysis and let it go into the citric acid cycle. So this occurs in the cristae of mitochondria. And this is just like little folds, okay? I'll show a picture in a second. But the whole purpose of the prep reaction is to take pyruvate that's a 3-carbon molecule, okay, and we convert it to a 2-carbon molecule. So we have to keep up with our carbons. So we are going to have an extra carbon left over, and this is going to leave as carbon dioxide. So we're just going to take our 2 carbons onto our next step, which is the citric acid cycle. So our acetyl group, which is a 2-carbon group, is going to attach to coenzyme A, and this is acetyl-CoA. Okay, carbon dioxide is produced because we have that extra carbon that we have to get rid of. We are going to have our electron carrier pick up 2 hydrogens, and this electron carrier is going to move on to the electron transport chain. Our acetyl-CoA is going to move on to the citric acid cycle. And this reaction does occur twice per glucose because glucose is split in half. So you have 2 pyruvate, and that's why it's going to occur twice. So for this little box, this is for 1 glucose molecule that this whole process does occur. Okay, so we're going to take our acetyl-CoA that we got from the prep reaction, and we are going to then move it forwards to the citric acid cycle. So the citric acid cycle occurs in the matrix of the mitochondria. The matrix of the mitochondria is kind of like the cytoplasm of a cell. It's just the gel inside of the organelle. So our 2-carbon acetyl-CoA group, okay, is going to combine with a 4-carbon molecule called oxaloacetate. You don't have to know the name, to produce citrate. Again, you don't have to know the name. All that you need to know is that acetyl-CoA goes in and then what comes out. But there's a whole process that takes place. So for every acetyl-CoA group, we're going to get carbon dioxide. Okay, we are also going to get electron carrier NADH and FADH2, and then we're also going to get some ATP. And again, this cycle turns twice per 1 glucose molecule because we have pyruvate that's split in 2 that creates 2 acetyl-CoA. So the citric acid cycle has to do twice because you have 2 acetyl-CoAs. So what goes in, we have our acetyl group that we get from the prep reaction. Our electron carriers, we don't really need to know that. We get carbon dioxide as a byproduct, but the main purpose of the citric acid cycle is going to be to create these electron carriers. And these electron carriers are then going to move over to the electron transport chain. Okay, so here we have our mitochondria. Remember, glycolysis is in the cytoplasm, prep reaction is in the cristae, which are the folds. Citric acid cycle is going to be in the matrix. And then we're about to talk about the electron transport chain, which also takes place in the folds, the cristae. So from the citric acid cycle, we got acetyl-CoA from the prep reaction, and the main product we're giving is NADH, FADH2, that's going to go to our electron transport chain, and we do make 2 ATP as a result of the citric acid cycle. Like I said, it is a cycle, so we end with what we start with. So if we look right here, we have acetyl-CoA that we got from our prep reaction, okay? One thing that when you're looking at this is just to keep up with your carbons, okay? That's what I keep up with. So we have 2 carbons right here, right? So acetyl-CoA, which is a 2-carbon molecule, is going to bind with a C4, a 4-carbon molecule, to create a 6-carbon molecule, which is citrate, right? 1, 2, 3, 4, 5, 6. So citrate is going to get rid of 1 carbon as carbon dioxide to become a 5-carbon molecule. 1, 2, 3, 4, 5. And when it gets rid of that carbon, it creates the electron carrier NADH. C5 is going to get rid of a carbon to create C4. When it does that, it gets rid of a carbon dioxide because you got rid of a carbon, but you're also creating electron carrier. C4, it actually is going to just rearrange to become a different C4. And when it does, we create this electron carrier FADH. And so we started with C4, we ended with C4, and then it moves on into the cycle again, picks up another acetyl-CoA, and goes along that way. So as carbons get released, we are creating electron carriers, but when we get rid of a carbon, we're creating carbon dioxide. Okay, that's all that we are going to do for this video.